This technique can be used to generate cell spheroids from individual and multiple cell types using a Lebanon CD platform, a pioneer system for the rapid generation of spheroids. The technique uses centrifugal force to deposit cells into designated microwells, allowing the sequential addition of multiple cell types for the production of diverse spheroids.
For CMS culture chip fabrication, first, make polycarbonate molds for the top and bottom layers of the culture chip by computer numerical control machining. Next, mix PDMS base and PDMS curing agent at a 10:1 ratio for 5 minutes, before placing the mixture in a desiccator for 1 hour to remove air bubbles. Pour the degassed PDMS mixture into the CMS culture chip molds, and place the molds into the desiccator for 1 more hour to remove any air bubbles. At the end of the second degassing, cure the mixture in a heat chamber at 80 degrees Celsius for 2 hours, before placing the chips in a vacuumed plasma cleaner with the surfaces to be bonded facing up. Expose the culture chips to air-assisted plasma at a power of 18 watts for 30 seconds. And bond the 2 layers of the chip in the heat chamber at 80 degrees Celsius for 30 minutes to increase the adhesion strength. Then, sterilize the CMS culture chip in an autoclave at 121 degrees Celsius.